Imagine a medieval castle, just under this massive, relentless siege.
Right. Complete lockdown.
Exactly. And for decades, the medical consensus has essentially viewed the human brain in exactly this way. You have the blood-brain barrier acting as this impenetrable moat, keeping almost everything out.
Yeah, that's the classic view.
And the tragic assumption has always been that if an invader specifically a metastatic cancer, somehow manages to swim that moat and breach those walls, the castle has absolutely zero internal defenses.
Right. It's a concept oncologists have historically called immune cold.
Immune cold?
Yeah. The belief was that the brain is just this barren wasteland where the body's immune system is either entirely locked out or, completely suppressed by the local environment. So if the cancer gets in, the immune system is essentially blindfolded.
But today, we are completely dismantling that myth in this deep dive. A recent study of 156 patients just proved that the medical world was, dead wrong.
It's actually staggering.
Right. The call isn't coming from outside the house. There are actually highly organized secret resistance movements operating right inside the castle walls.
Oh, absolutely.
We are unpacking a groundbreaking preview article published in Cancer Cell by Shangliang Yuan and DOU, and they're detailing a monumental new study by Jasovitz and colleagues on breast cancer brain metastases, or BCBMs.
And they didn't just find a few lost immune cells wandering around. They found intricate, highly lethal biological networks fighting back.
So if the prevailing thought was that this castle had no internal defenses against invaders, how did this study of 156 patients prove there are actually secret resistance movements operating inside?
Well, what's fascinating here is the sheer scale and the technology they used. They brought a massive amount of firepower to the table. I mean, we're talking tissue cytometry, single nucleus RNA sequencing, and spatial transcriptomics.
Let's break that down for everyone listening, because that sounds like a lot of jargon.
All right, sorry. So historically, if you wanted to analyze a tumor, you'd take a biopsy, grind it up, and sequence the genetics. It's basically the equivalent of throwing a finished Lego castle into a blender to figure out what color bricks were used.
You know what's in there, but you lose all the architecture.
Exactly. You have no idea how it was put together. So instead of the blender approach, this team used spatial transcriptomics.
Which is like a highly sophisticated Google Maps for biology, right?
That's a perfect way to describe it. It doesn't just sequence the genetic diary of individual cells. It uses these tiny spatial barcodes to tag exactly where that cell is located on a microscopic tissue slide.
So it tells you the cell's zip code.
Yep, its zip code, who its neighbors are, and importantly, who it's actively communicating with.
So they literally map the battlefield. And when they mapped these 156 brain tumors, they didn't just find some generic disorganized immune response.
No, not at all. They discovered two completely distinct, prognostically favorable niches, like two separate ecosystems.
And if you or a loved one are facing this diagnosis, understanding these hidden fortresses changes everything. It's the difference between a generic treatment and a precise biology-driven intervention.
Absolutely.
Okay, let's unpack this. We have to start by looking at the first major ecosystem they mapped, which is the TRM-like niche. What are we looking at directly inside the tumor itself?
The clinical headline here is intratumoral CD8 positive T cell abundance. Basically, having a lot of these specific T cells inside the tumor is an independent, favorable prognostic factor. It means patients live longer.
Let's visualize how these cells operate. Because if your normal circulating immune cells are like traveling infantry soldiers just patrolling the blood highways, these specific CD8 positive T cells are deeply embedded undercover agents.
Right. The researchers call them tissue-resident memory cells, or TRMs.
And they live right inside what the mapping showed as tumor islands, which are these dense clusters of epithelial cancer cells.
They are completely entrenched. And we know they are entrenched because of a specific marker they express called CD103. Yeah. You can picture this CD103 protein as a literal grappling hook. It binds directly to the cancer cells, anchoring the T cell in place so it doesn't get pushed out of the tumor microenvironment.
So they throw the grappling hook, anchor themselves deep in enemy territory, but they aren't just hiding there. They are heavily armed, right?
Oh, they are carrying a serious payload. They co-express cytotoxic molecules, the most important one being granzyme B.
I've always thought of granzyme B as a biological grenade.
That's actually a really accurate way to look at it. When this anchored CD103 positive T cell recognizes the cancer cell it's attached to, it essentially pulls the pin and injects the granzyme B directly into the invader.
Wow.
Yeah, and this enzyme physically cleaves proteins inside the cancer cell, triggering apoptosis. It forces the tumor cell to commit suicide. And this combination, the CD103 grappling hook plus the granzyme B grenades, is directly linked to local tumor control.
That's incredible. And because of the spatial mapping, they saw these anchored assassins are totally segregated from other immune cells, right?
Right. Yes, specifically, they're segregated from another group of CD8 positive T cells that express GCMK, or granzyme K.
What's the difference?
Well, the GCMK positive cells are stuck on the outside. They are in the peritumoral regions, often trapped near suppressive macrophages, specifically MRC1 positive macrophages, which just shut them down.
So the elite assassins with granzyme B are physically inside the keep, while the GZMK guys are stuck outside the moat dealing with the guards.
Exactly. And the researchers proved how effective the inside guys are using ex vivo organoid-killing assays.
Oh, I love this part. They literally watched the warfare play out in real time.
Yeah, they took actual tumor tissue from patients, grew these miniature 3D tumors organoids in a Petri dish, and watched under a microscope. And they saw that the CD103 positive cells killed the tumors significantly better than their CD103 negative counterparts.
But this brings up a massive contradiction, doesn't it? If these TRM cells are so great at killing, why aren't they winning the war? Why is breast cancer that spreads to the brain still so deadly?
Because the tumor fights a war of attrition. As these T cells attack, the tumor learns to manipulate the immune system's own safety mechanisms.
The molecular breaks.
Right. Immune checkpoints like PD-1 and TIG. The researchers found that these embedded TRM cells highly co-express PD-1 and TG.
And the body needs those breaks normally, right? So the immune system doesn't go hyperactive and destroy healthy brain tissue?
Precisely. It's a necessary safety feature to prevent autoimmune damage. But the cancer cells figure out how to press those brake pedals. The tumor effectively slaps handcuffs on our undercover agents. The T cells become what we call exhausted.
So they are anchored on site, fully armed, but completely immobilized.
Yes. And that's why the researchers nominate a dual blockade blocking both PD-1 and TIGIT as a rational therapeutic strategy. If you block the brakes, you take the handcuffs off these assassins.
Okay, so bullet point summary for you listening. 1, the brain isn't strictly cold. 2, embedded CD103 positive TRM cells are our localized assassins using biological grenades. And 3, blocking TGAD and PD-1 helps take the brakes off these assassins.
That perfectly sums up the TRM niche.
So think about it. If you were designing a drug, would you focus on bringing new troops into the brain or just taking the handcuffs off the ones already there? It totally changes the paradigm.
It really does. But as powerful as those localized assassins are, hand-to-hand combat is only one part of the resistance.
Right, an embedded assassin is great for localized damage.
Yeah.
But to wage a full-scale war, you need a base of operations. Which leads us perfectly to the second, entirely distinct immune niche.
Yes, the tertiary lymphoid structures, or TLS.
Pop-up bases. I love that visual. Where exactly are they setting up camp?
Well, the spatial maps show they are highly organized perivascular lymphoid aggregates.
Perivascular meaning built aggressively around the blood vessels in the brain.
Exactly. They aren't just random clusters of cells, they have deliberate architecture. It's literally an immune base camp built right in the metastatic environment.
Let's walk through the architecture of that base. Let's call these the T&T of brain defense, right?
Okay.
TRM is the resident muscle, and TLS is the local station.
I like that. So, the local station, the TLS, is incredibly complex. It contains distinct B cell zones and plasma cells.
Which are the body's munitions factories, pumping out antibodies.
Right. And to coordinate that, you have mature dendritic cells, specifically DCLMP positive dendritic cells.
So those are like the intelligence officers grabbing tumor fragments and bringing them back to the base.
Exactly. And they present that intel to a highly specialized group of CD4 positive T cells. The researchers identified T follicular helper cells, or TFH-like cells, and central memory subsets in these structures.
So the scavengers hand the intel to the generals, the CD4 cells, who then tell the B cells what antibodies to make. It's a literal assembly line of immunity.
It really is, and here is where it gets really interesting.
Oh, lay it on me.
You would assume that a patient doing well would have both of these systems working together, right? Like the base camp is coordinating the embedded assassins.
Yeah, that would be the logical assumption.
But the spatial analysis proved the exact opposite. TLS positive and TRM high phenotypes overlap minimally.
Wait, really? Minimally?
Yeah, they operate almost totally independently from each other.
So a patient might have the embedded assassins, or they might have the manufacturing base camps, but rarely both. Why is that such a crucial finding?
Because it tells us these are independent routes to favorable immunity. They aren't just two sides of a generic inflamed state.
Meaning there isn't just one narrow path to fighting brain cancer.
Exactly. From a clinical perspective, this is huge. It dramatically expands the biomarker-defined population for cranial immunotherapy. We have multiple targets now.
Okay, bullet points for part 2. 1. TLS are organized immune base camps around blood vessels. 2. They contain B cells, DCLMP positive dendritic cells, and CD4 positive T cells. And 3. They operate totally independently from the TRM assassins.
Spot on.
So, audience question. If these two systems work independently, could a future therapy trigger both simultaneously in a patient?
That is the absolute frontier we are striving for.
But logically, if TLSs are these amazing pop-up base camps, who exactly is building the scaffolding for them? The immune system doesn't just float in empty space.
No, it needs structure. And this brings us to the myeloid layer, the architects and the suppressors.
Because historically, brain macrophages and microglia have a terrible reputation. Right?
Yeah,
They are predominantly immunosuppressive.
they are. The tumor is incredibly manipulative. It hijacks these cells, specifically the MRC1 positive and Marco positive tumor-associated macrophages.
The suppressors.
Right. They secrete chemicals that put the attacking T cells to sleep and help the tumor grow. They become traitors.
But the study found heroes hidden among the traitors. Who are the good guys?
The researchers identified specific favorable subsets, HP positive and CCL3 positive microglia-derived TAMs.
TAM-MGs. So these are native brain immune cells actually fighting for us.
Yes, and they have independent prognostic significance. Their superpower is their extremely high expression of HLA-DR.
Let's visualize HLA-DR. Think of it as a massive glowing billboard on the outside of the cell, right?
Exactly. The macrophage consumes a piece of the tumor and sticks the fragments right onto that HLA-DR billboard. It proudly displays the enemy so the rest of the immune system knows what to attack.
The ultimate biological wanted poster.
Yeah. And spatially, these HLA-DR high myeloid states were heavily enriched right inside the TLS domains.
Inside the base camps.
Right. And they do more than just hold up signs. They are closely associated with CXCL13.
CXCL13, which acts like a chemical beacon, right?
Yes. They broadcast the CXCL13 scent trail to physically draw B cells and T cells into the area.
So let me synthesize this. These specific myeloid cells aren't passive bystanders. They're the architects. They're the antigen-presenting scaffold holding the TLS base camp together.
It's exactly that. It reveals the chaotic heterogeneity of the tumor microenvironment. Mere micrometers apart, you have macrophages protecting the tumor and others pouring concrete for an immune fortress to destroy it.
Quick summary of part 3. 1. Not all brain macrophages are bad. 2. HENKI positive and CCL3 positive subsets are our heroes. And 3. The CXCL13 expressing cells provide the physical scaffolding for our local immune stations.
Perfectly stated.
So think about this. How wild is it that cells originating from the brain's own native immune system, the microglia, can be co-opted to either protect the tumor or build fortresses to destroy it?
It's a literal cellular civil war.
It's mind-blowing. But okay, now that we know all the players on the board, the assassins, the stations, the architects, how do we actually use this to save lives? So what does this all mean for the patient?
This is the most critical clinical takeaway from the entire preview article. You cannot infer the immune state of a brain metastasis from the matched primary breast tumor.
Meaning you can't just look at the original breast biopsy.
Right. Standard practice often relies on that primary tumor to guess what's happening in the brain. But the authors point out that while TRM and TLS signatures were enriched in basal-like primary tumors, they were actually found across all BCBM molecular subtypes in the brain.
Including luminal breast cancer, right?
Yes. In the breast, luminal cancer is often immune cold. But in the brain, it can evolve to spark these massive pop-up base camps.
Wow. So the brain lesion evolves distinctly.
Exactly. Patient selection for cranial immunotherapy requires assessing the intracranial lesion directly. You have to test the brain tumor itself.
That is a massive paradigm shift. But to actually cure this, The expert authors outline 3 major unanswered questions, right? The 3 great unknowns standing in our way.
Yes. The first gap is the nature versus nurture of the TRM cells. Are they pre-primed at the breast and travel to the brain, or are they born locally in the brain's unique metabolic environment?
Systemic expansion versus local reinvigoration.
Exactly. If they travel, we need systemic drugs. If they are born locally, we need drugs that cross the blood-brain barrier.
Makes total sense. There's a second unknown.
The TLS trigger. What specific stromal signals cause these base camps to form in a site of immune collapse?
Because if we know the trigger, we can artificially induce them in TLS-negative patients, right? We force the body to build a base.
That's the holy grail. And the third gap is the myeloid gatekeeper. Are those HAM-P positive and CCL3 positive TAMs actively stabilizing the bases?
Like true architects.
Right. Or are they just showing up to an already inflamed area? We need to know if they are laying the foundation or just, arriving after the house is built.
So final bullet points. 1, you must test the brain tumor itself, not just the primary breast tumor. 2, we still need to figure out where these immune cells originate and how to manually trigger their formation.
That covers the clinical mandate perfectly.
This entire deep dive has been a massive paradigm shift. We are moving from viewing brain metastases as strictly cold to understanding them as intensely complex and capable of harboring multiple independent resistance networks.
And knowledge of this complexity is our ultimate weapon. We have to move away from stagnant therapeutic options. We're moving towards highly precise, biology-driven immunotherapy that actually respects the unique environment of the brain.
It's incredible. So I want to leave you, the listener, with a final provocative thought. If the brain, the most heavily guarded, historically immune-privileged organ in the body, can learn to build these complex immune fortresses to fight cancer, what other rules of human biology might we be completely wrong about?
That's a great question.
Right. If tumors can evolve so differently in the brain than in the breast, how much of our current medical rulebook needs to be rewritten based on where disease lives rather than just what it is?
It really makes you question everything we thought we knew.
It absolutely does. Thank you for joining us on this deep dive. Keep questioning the consensus, and we'll see you next time.